Imagine a world teeming with life, all contained within a simple glass flask. This is the reality of plant cell suspension culture, a fascinating technique that allows scientists to grow plant cells in a liquid nutrient broth. This method, a cornerstone of plant biotechnology, offers a unique window into the hidden lives of plants. It allows us to study their growth, manipulate their genetics, and even harness their medicinal properties, all in a controlled laboratory setting. This approach is revolutionizing how we interact with the plant kingdom. It opens doors to new discoveries and applications, from developing novel medicines to creating sustainable solutions for agriculture and industry. Join us as we delve into the intricacies of plant cell suspension culture, exploring its potential to reshape our world. Before we delve into the process, let's consider the essential ingredients that make this cellular magic possible. First, we need the plant cells themselves. These are often derived from a callus, a mass of undifferentiated cells that forms when a plant is wounded. The callus, much like a plant stem cell, has the remarkable ability to develop into different cell types, making it an ideal starting point for our culture. Next comes the culture medium, a carefully concocted liquid broth that provides all the nutrients the plant cells need to thrive. This life-giving elixir contains a precise blend of sugars, vitamins, minerals, and plant hormones, each ingredient playing a crucial role in cellular growth and development. Finally, and perhaps most importantly, we need to maintain a sterile environment. Just like our bodies, plant cells are vulnerable to contamination by bacteria and fungi. To prevent these unwanted guests from taking over our culture, we employ strict sterilization techniques, ensuring that our plant cells have the pristine conditions they need to flourish. With our ingredients prepped, we can begin the process of establishing our plant cell suspension culture. The journey starts with carefully selecting a piece of friable callus, chosen for its ability to easily break apart into individual cells. This callus is then introduced into a flask containing our nutrient-rich culture medium. To encourage the callus to disperse and the individual cells to multiply, the flask is placed on a shaker, gently agitating the culture. This movement ensures that the cells are evenly distributed and have access to the nutrients and oxygen they need to grow. This incubation period is crucial, allowing the cells to adjust to their new environment and begin to proliferate. As the culture grows denser and the nutrient levels in the medium deplete, it's time to subculture. This involves transferring a small portion of the cell suspension into a new flask with fresh culture medium, providing the cells with a renewed supply of nutrients and space to expand. This cycle of growth and subculturing can be repeated indefinitely, creating a continuous supply of plant cells for research, production, or other applications. Section 4. Applications. Unlocking nature's potential. The ability to grow plant cells in a controlled environment opens up a world of possibilities. One of the most promising applications lies in the production of valuable secondary metabolites. These are compounds, often with medicinal properties, that plants produce for defense or to attract pollinators. Plant cell suspension cultures can act as miniature factories, churning out these valuable compounds in quantities that would be difficult or impossible to obtain from whole plants. Another exciting application is in genetic engineering. By introducing specific genes into plant cells in culture, scientists can create plants with enhanced traits such as resistance to pests or tolerance to drought. These genetically modified plants hold immense potential for improving crop yields and ensuring food security in a changing climate. Beyond these applications, plant cell suspension cultures are also used in the production of synthetic seeds, tiny capsules that encapsulate plant embryos and nutrients, providing a convenient and efficient way to propagate plants. This technology has significant implications for conservation efforts, allowing for the long-term storage and distribution of rare and endangered plant species.